All right, so this lecture is on something that's very relevant right now, I believe, about viruses and prions. Now, obviously, viruses are important. We can tell that right now. Viruses are what we would call an indiscriminate infection or an indiscriminate pathogen. Meaning it doesn't really matter who you are. You can be rich, you can be poor, you're all equally as susceptible. When it comes to bacterial and fungal infections and even protozoal infections, it tends to be more discriminate, meaning that people who are in certain economical classes will be more likely affected by a bacterial disease or a fungal disease or even a protozoal disease, while other economical classes will not. But viruses don't care. Viruses will infect everybody. And viruses, not only because they can do this, infect everybody, they really have a toll on our history, our culture. In fact, wars have been won and lost just based on viral infections. If you ever wonder to yourself, why is it that the United States ends at its northern border with Canada? Why does it end there? Why didn't George Washington and all his armies just continue all the way north up through Canada? Well, they tried to. In fact, what happened is George Washington's army had a town in Canada, a city in Canada, surrounded. And what the Canadians did, really the British did at that point, was they were held up in this town. They were barricaded in. So what they did was they took prostitutes from the town, infected them with smallpox, and then let the prostitutes out into the Washington's army that was basically keeping them prisoner in that town. And within a matter of days, George Washington's army became sick with smallpox, and they had to run back because otherwise they would all die. I mean, that's amazing. So viruses, they are really important when it comes to human civilization. Entire countries are devastated by viruses. When Europeans came to America, variola almost killed everyone in America, all Native Americans. Smallpox, it kills in the 20th century, the century that we were all born in, killed an estimated 300 million individuals. That is the population of the United States. It kills indiscriminately. It kills emperors and queens and kings, and it doesn't matter who you are. Just to give us an example of some history when it comes to the history of viruses, is that smallpox propelled the establishment of Christianity in the Latin American world. How does a virus establish a religion in an entire continent? Well, this has to do with what happened in Europe during this time. So during the 15th and 16th century, smallpox was endemic in Europe, meaning that it was always there. A lot of people had it all the time. And when you got smallpox, if you were old, you died. If you were young, you probably died. But if you were middle-aged, sort of like, a young adult and an early a midlife adult, you would probably survive. The problem with this is that these people who survived, they end up serving in the army, in the military. And then in comes in these conquistadors. Conquistadors were from Spain, like Cortez and Pizarro, and they brought these soldiers with them that were infected with smallpox and measles. And they brought it to the New World, which was the Americas. And the Americas never had smallpox and measles before. And what that means is that Native Americans never had an evolutionary pressure to create any sort of resistance in their population against these diseases. So as the conquistadors came to America, bringing these new diseases, well, it essentially spread like wildfire through the Native Americans. And all the people, like the Aztecs, that were dying in such great numbers that we're still finding towns buried in the forest, in the jungle, today. I mean, imagine how quickly you have to have a population die where major towns and civilization centers are just lost to history. And we're only just finding them. It's amazing. And if you think to yourself, you know, all these people are dying, what are the Aztecs going to do? Well, what would people do in the case when a lot of people start dying, a lot of people of your culture? Well, we can see what's happening now. A lot of people are praying. They look to their religious deities and things like that, like Quetzalcoatl from the Aztecs. But it wasn't helping them. They kept on dying. Now, what's crazy about this history is that the Aztecs had a prophecy that said that gods would come to them over the ocean riding these great beasts. Now, horses didn't exist in America before the Europeans. And when the Europeans came, they were riding horses. And the Aztecs took that as if they were gods. So instead of praying to their God, they started praying to a Christian God, because those Europeans, well, they weren't surviving this epidemic of disease and death. So it took to them that, hey, the Christian God must be better than our God because our God is not saving us. And to think, you know, how big of an impact this was on the Latin American society, Latin American culture, is if you think Latin Americans and how they hold their religion, it's very, very strong. And all of this because of a virus. Now, viral diseases have been documented for ages, even bacterial and fungal diseases, documented for ages. What we're looking at over here is a sample from a tomb, a burial ground. And it's all these fruits and these seeds, like almonds and things like that. All these things, they have a lot of vitamin C. And guess what? Even the ancients knew that vitamin C would help them actually recover from a cold better. The first seasonal flu that was ever documented was first described by Hippocrates. We all know Hippocrates now, Hippocratic Oath. In 412 BC, he wrote in the book of epidemics, yes, there was a book of epidemics, and he described a influenza-like winter illness that was called the fever of Perinthus, or the cough of Perinthus, which is where this fever was occurring. Now, not all virus outbreaks are the same. Well, there's seasonal flu, seasonal colds all the time. And we don't have to basically pack things up and do off-site instruction every time there's a cold around. But every so often there is a change in the virus where it jumps from one organism to another organism, from one host to a completely different host. And this type of disease is known as a zoonosis, or a zoonotic disease. And when this happens, this is what's going on. We believe that this new strain of coronavirus, COVID-19, originated in an animal, which then made the jump to a human. And because it was new to humans, it spread very rapidly. In fact, the spreadability, something called the R0 of the COVID-19, is something around 2.5 to 3, which means that one individual infected with COVID-19 can infect 2.5 to 3 other people. Let's just compare that with other types of viruses. The 1918...
Can you repeat what that word was? R0?
R0, or RO.
Thank you.
Yep, the typical, well, not typical, but the 1918 Spanish flu, which killed a massive amount of people 100 years ago, had an R0 value of about 1.3 or 1.5. That's nothing in comparison to this virus that we're dealing with right now. So what is it that causes viruses like seasonal flu or seasonal colds to become something more dramatic, pandemics? Well, it has to all do with what happens during the replication of the viruses. As a virus replicates, it slowly builds up mutations. And after about 1 year, the mutations have changed the virus just enough that our immune system has trouble recognizing it again, i.e., you got the seasonal flu last year, well, you're going to get it again next year. That's because as the year has gone by, the flu virus that is going around in the populations is slowly changing, where our immune system no longer recognizes it. This slow change in a virus that comes back every year, this is known as antigenic drift. Antigenic drift. So we have this slight change in how the virus appears, which causes our immune system not to respond. So here we see on the surface of a virus the structure called hemagglutinin. Don't worry about what the word means yet. But what we notice is the hemagglutinin from the previous year has more of this pentagonal structure, but then next year the structure has changed. Because that structure has changed, that means that our antibodies don't really recognize it as much as it did before, or from the last time we saw it. So that's the mechanism by which seasonal flus and seasonal colds come about. Why you get a cold every year, why you get a flu every year. But when it comes to these more dramatic versions of the virus, how do they become pandemics? Well, let's look at influenza, for example. This can be the same thing for the coronavirus. Influenza does not only infect humans. It infects a variety of strains of viruses. It infects a variety of organisms, such as pigs, birds, bats, you name it, seals. Now, each type of influenza virus doesn't only just infect the one organism, like pigs by itself, but it can also infect pigs and humans. The birds can infect birds and humans. Bats can infect maybe bats and humans, or maybe bats and pigs. So this is what's known as a host range. A virus can infect a few different types of hosts. Now, on occasion, what happens is that a variety of strains of the flu infect a single host, meaning that here you have a pig flu, and here you have a bird flu. And the pig flu and the bird flu both infected a pig. And inside this one host organism, it creates a recombination of the two viruses, the pig virus and the bird virus. And as a result, produces a conglomeration or a chimera of the two organisms, or of the two viruses. A chimera means that you have a mashup of two organisms, like a griffin is a mashup between a bird and a lion, right? So you get this mashup of the two viruses, virus A and virus B. These two viruses come together, and now if they can infect humans, well, we've never experienced a pig-bird flu before. And that means that our immune system cannot recognize the virus at all. In the antigenic drift, our immune system has trouble recognizing the virus every year. Doesn't mean it can't do it at all. But when it comes to antigenic shift, this new combination of virus from the different strains makes it so that our immune system has absolutely no defense against it. So Holden asks, is that what swine flu was or bird flu was? Yes, that is what swine flu was and bird flu was. It was a recombination that allowed a typical swine flu virus that infects pigs only typically to now also infect humans. That is also what happened with birds. A bird flu that typically only infected birds was able to make a jump by a recombination, a chimera of bird flu and something else, to now infect humans. So this is what's known as antigenic shift. And antigenic shift produces pandemics. And that's what we think is happening now. So here we have an example of this antigenic shift. So we have these different types of flus. You may have heard of these names before, such as H7N7. H7N7 is the bird flu. H1N1, that's swine flu. The typical human flu is known as H3N2. This is the flu that comes back every year. Now, each of these flus has a host range, meaning that the bird flu doesn't only infect birds, it can also infect pigs. The human flu, it doesn't just infect humans, it can also infect pigs. So what you create as a result of this is sort of this mixing pot in this one organism, the pig, where this pig might be infected with bird flu, pig flu, and even human flu. So you have these 3 different flu viruses in this one organism, this one melting pot, the pig. And as a result of this, all 3 different strains of the flu mix together. And what we get is this chimera. This chimera consists of part human flu, part bird flu, part swine flu. And it comes back to us, and our immune system can't handle it. It has absolutely no defense. And because it has absolutely no defense, the virus can replicate uncontrolled and therefore cause a lot of serious actions and even be very, very infected. It can spread very quickly, become very contagious. So in 1918 was the last time that we had a major flu pandemic. There were a few of the flu epidemics, such as swine flu and bird flu, but the last flu pandemic was in 1918. So in 1917, so in 1917, there was a seasonal flu, and it infected a lot of people. But then it suddenly went away. And then 1918 comes around, and the flu that next year, in 1918, infected nearly 500 million people in the world. That's almost half the population, or a third of the population of the planet at that time. It was about 1.8 billion people. And within that time, it killed 10% of the people who were infected, about 50 million deaths in just a matter of months. Now, if we look at what's happening now with the coronavirus, there is about 250,000 people documented to be infected with this coronavirus, with about 11,000 deaths. So while not as severe, this 10% that we see in the Spanish flu, it's still a pretty severe infection. About 5% so far, what we see is that people are dying from the COVID-19. Now, what we're trying to do, the reason that we are self-quarantining, social distancing, not going to work, staying off the trains, is because we do not want to overwhelm our hospitals. You may have heard of this, it's called flattening the curve. Because if we take a look back in history in 1918, when this happened, it happened over a matter of weeks to a matter of just a month and a half. And so many people got sick so quickly that there was nowhere to put patients. So what they had to do was they had to commandeer public buildings like dormitories. You may have heard that many people have had to be kicked out of their dorms. This is because dormitories become makeshift hospitals, even high schools. If we look at this picture over here, you know, what is this building? We see a lot of beds here, but it doesn't look like a hospital. That building is a gymnasium from a high school, and it is packed filled with cots with patients in it. This is what we're trying to prevent. We cannot prevent the spread of the disease, but what we're trying to do is we're trying to make it so that the time it takes for one person to infect another is delayed. And by delaying the spread and spreading it out over time, our hospitals can handle this. So if we just take a look at a quote from one of these military physicians during this time, what he said was, "It is only a matter of a few hours until the deaths comes. It's horrible. We have been averaging 100 deaths per day, and that was in one of these facilities. Just last night, Spain experienced over 250 deaths in one night. This was so dramatic that so many people died, they had nowhere to put them. They literally had to just leave the dead bodies in houses. And during this time, the life expectancy of an American, a US citizen, dropped by 12 years. During this time, your life expectancy was actually to 50, and it dropped to 38 years old. I'm pretty close to that age. That would be your life expectancy. That's astounding. So where were viruses originally discovered? I mean, we knew about their presence since ancient times. Hippocrates knew about it. The ancient Egyptians knew about them. They tried to use citrus, vitamin C to ease the symptoms. Where virology, the study of viruses, kind of starts is in 1887 with a scientist called Dmitry Ivanovsky. And what he was working on was a virus or a disease called tobacco mosaic disease. And what tobacco mosaic disease does is it takes a tobacco leaf, like we see here, a nice healthy tobacco leaf, and it causes to form all these little spot marks on it. These spot marks look like a mosaic. It's a mosaic of spots. So they call this the tobacco mosaic disease. And what Ivanovsky showed is that if he just took the sap of these infected plants, that he could give another plant this same disease. So the sap itself was able to spread the disease. And what he called this was a contagium vivium fluidium, which meant that it was a contagious fluid that was alive for some reason. But Ivanovsky went one step further than just showing that the sap can spread the disease. He found that if he passed the sap, the contagium vivium fluidium, through a filter, that even the filtered material could still spread the disease. So let's look what a Chamberlain filter is. So what we're seeing over here, this is a Chamberlain filter. And what a Chamberlain filter is, it is unglazed ceramic material. So here we see the unglazed ceramic material. If you've ever touched unglazed ceramic material, you'll notice that it's very rough, and that's because the fact that it's unglazed means it's very porous. Now, what the Chamberlain filter was used for in science was to filter out cells. It was small enough to filter out even bacterial cells. So he passed this contagious fluid through the Chamberlain filter, and if it was cells that were causing the disease, well then the filtrate shouldn't pass the disease on to another organism. What he discovered was that even if you filtered out all the cells from the contagious fluid, it would still pass on the disease. So like I was saying, these filters, these Chamberlain filters, are made of unglazed porcelain. And these unglazed porcelain filters, they contain pores that are too small to permit bacteria to pass through. So what this did was it can create a new experimental approach into demonstrating this new type of infectious agent. And what we call this infectious agent is a virus. And what virus actually means is toxin, because what they thought it was some sort of toxic material that was not living cells. Now, the thing with what Ivanovsky did was that he was not able to grow his virus or this toxin in culture from the infected sap. And because he couldn't do this, he could not satisfy one of the essential components of Koch's postulates, which we'll talk about in a couple weeks. But and because of this, because Ivanovsky could not isolate and grow his virus, people said, well, no, maybe his filters are broken. Maybe it's not a living thing. Maybe it's a toxin. So this was defeated, these second guessings of Ivanovsky's filter contagious fluidium vivium was by a Dutch microbiologist, Martinus Bergenek. In 1898, what Martinus did was he demonstrated that the sap, if it were a toxin should dilute every time you give it to a new host. So you take it from the original host, you give it to a new host, you then allow that new host to become diseased, which should take up some of that toxin. And if you give it to another host, well, the time it should take for that disease to come on should be slower. And if you get it to another host from that previous host, it should diminish over time. What he saw was that if you took the sap from the infected plant, you gave it to a new plant, that new plant became diseased. If it were a toxin, meaning that now this toxin is used up here in this plant, if you were to take that and now give it to another plant, it should stay healthy because all the toxin was used up to make this plant sick. But instead, what Martinus showed was that if you took the sap from the originally infected plant, gave it to a new plant, the new plant got sick, took the sap from that, gave it to another plant, that plant got sick, took the sap from that third plant, gave it to a fourth plant, that plant got sick, from the fourth plant to the fifth plant, that plant got sick as well. A toxic chemical could not do this because every time you go from one host to another, you're using up that toxin and therefore it dilutes out. So there should be a point, if it were a toxic chemical, where it should no longer be toxic anymore because it's too dilute. What Martinus showed by this experiment is that the toxin was able to replicate, thusly replenishing its toxicity. So it was able to go from host to host to host to host and continuously infect new hosts from a previous host. So this established two fundamental properties of this contagious fluid. Number one, they're smaller than bacteria. And number two, they require living tissue to propagate. This is why Ivanovsky failed. Ivanovsky could not get that contagious fluid to replicate in a culture. That's because he didn't realize that it needed tissue to propagate. Now, the story is still incomplete here because they weren't able to actually see whatever this material was. So what they refer to it as is at this point a filterable agent, meaning it can pass through a filter that should normally filter out cellular pathogens, but it can still cause disease. The first time that we actually saw a virus was in 1938, where an electron microscope was created, I believe by the IBM company or someone like that, or Rockefeller University. So in 1938, the first electron micrograph was used to identify what was causing that tobacco mosaic disease. And what it was discovered was that the filterable agent was actually these particles. And what it was come to be known as tobacco mosaic virus, or TMV. The reason they called it a virus was not because of, virus is a virus. No, it's because virus in Latin means poison. So this was often a term used when describing that filterable agent as a poison. So Jamie asks me if infectious agents then lose value as they carry from host to host. Well, no, Jamie. A toxin, as in a chemical, will lose its toxicity from host to host. But an infectious agent, which can replicate, maintains its infectivity because it can replicate, while a chemical toxin cannot. So that's what Martinez showed, that it wasn't a chemical agent because a chemical agent should lose its toxicity over hosts. But here, this tobacco mosaic virus or disease was able to maintain its infectivity from host to host to host to host. So by Wendell Stanley identifying those filterable materials, the tobacco mosaic virus, it allowed finally for us to start to understand what viruses actually are, such as what is the chemical composition of a virus and what's the structure of a virus. So question is, are viruses alive? Well, if we look at what a virus is and compare it to a living organism, a living organism carries out metabolic processes. We all know now what metabolic processes are. This is where enzymes are used to drive some sort of biochemical reaction. Well, viruses by themselves don't do that. Living organisms, they maintain homeostasis, meaning they maintain a constant internal environment. Well, viruses don't do that. Living organisms, they have the ability to reproduce, making copies of themselves by themselves. Well, viruses can't do that by themselves. They need someone to help them. So defining a virus as a living organism is very difficult. We can't, because viruses outside of a host, outside of a living cell, are completely inert, meaning they can't really do anything. And it's only once they're within inside the host, the cell, that the viruses become active. But it's really the host cell that's doing all the stuff for the virus. So one of the terms that's sometimes used, and I really hate it, is a virus is considered a non-cellular form of life. I really hate that term because it really smacks in the face of what living things do. I don't like that term. Instead, a virus, for me, is a virus. They fall into their own category. So the exact definition of a virus is that they are obligatory intracellular parasites, meaning that they have to, obligatory means they have to be inside the cell. And once they're inside the cell, they feed off the cell. They use the cell for their own gain, and the cell itself becomes harmed. Now, if we look at their composition, viruses contain DNA or RNA. They never contain both. They either have DNA or they have RNA. Surrounding this DNA or RNA is a protein coat. Sometimes the protein coat is surrounded by an envelope, which is made out of plasma membrane. Viruses have these structures called spikes. The spikes are used to determine what type of host that cell will infect. And most viruses, they only infect specific types of cells in one host. So if we take, for example, the flu infects respiratory epithelia. It infects respiratory epithelia regardless of what host it's in. If it's in the pig, the bird, or humans, it's always the respiratory epithelia which they target. Now, like we said with those flu viruses, they don't just infect one type of animal. They can infect multiple types of animals. And we call this the host range of a virus. The host range is the spectrum of host cells or hosts that a virus can infect. Some viruses, they can infect only one specific cell type of one specific host. Others can infect multiple types of cell types in multiple types of hosts. If a virus infects only bacteria, we call that type of virus a bacteriophage, or simply a phage. So what determines the host range? The host range of a virus is determined by the attachment sites and cellular factors of the host. So on the surface of a virus, there are these chemicals, and they interact with receptors on the surface of a host cell. If we look at bacteriophage, for example, those are the ones that infect only bacteria. They look for things that are on the surface of a bacterial cell. Things such as the cell wall itself. The cell wall behaves as a receptor for the bacteriophage. It can also be the fimbriae or the flagella. So they're looking for these surface structures in which they can attach to. And those surface structures which they can attach to are going to be very specific for that virus. In the case of animal viruses, it's going to be looking for certain structures that are found in the plasma membrane. So viruses, they only infect certain types of hosts. And within those hosts, they may only infect certain types of cells. And the reason for that is because they're looking for specific receptors on those cells contained within inside those hosts that they can attach to. Therefore, if an organism does not have that specific receptor for that specific virus, well, that host will not get infected, which is why humans don't get infected by bacteriophage. We don't get infected by bacteriophage because we don't have the receptors that the bacteriophage is looking for. So viruses look for specific receptors. What about what they look like? Well, viruses generally come in 3 different types of shapes. One shape is called the icosahedral. The icosahedral is a 20-sided structure. So here we're seeing the icosahedral. The other name for icosahedral is polyhedral. What it's made up of is 20 flat triangles. Another structure is called the helical. In the helical structure, the virus looks like a tube, what we can see here, like the tobacco mosaic virus, like we saw in that previous image. The last type of viral structure is called complex. Complex structures are literally that. If we look at this complex virus, we can't really call it polyhedral and helical. It's complex. It has all these various structures on it. Now, complex viruses are can only be phages. All complex viruses are phages. And what they have is this structure called a head at the top, followed by a tail, which then has these tail fibers coming out of it. We're going to talk more in detail about this structure. So there are 3 types of viral morphologies: the icosahedral, the helical, and the complex. When we look at an individual viral particle, we call that a virion. And a virion generally consists of 2 things: nucleic acid and protein. So a virion, its nucleic acid can be either DNA or RNA, never both. Can anybody guess why it could never be both? Why is it that a virus will either have DNA or RNA? Can anybody take a wild guess at that?
Because it's not that complex and it doesn't produce, as a virus, it doesn't really produce anything. It just...
Yeah, but why would it only have DNA or RNA? No, it's not so that it may be transcribed. Why does it only have RNA or only DNA? Okay, you're all going into a different direction. No. Well, no, it's not because it's not a living organism. It's not because they need to replicate. They can replicate with DNA or RNA. It's because the viral particle is too small. It's too small to have both. So by having one nucleic acid, you have all nucleic acids, right? The viral particle is just too small to have both types of nucleic acid. Let's just recall from our DNA lectures. If we have one strand of DNA, let's say it reads A, T, G, C, C, A, well, all you need is that one strand. And from that one strand, you can make the opposite strand of DNA, T, A, C, G, G, T, or you can make your RNA, U, A, C, G, G, U. What about if you just had RNA? If you had RNA, UAAGC, if you had RNA, well, you can make DNA from RNA. You can, because all you have to do is make the complement. So this would be A, T, T, C, G. By having just one strand or just one type of nucleic acid, it means you have all types of nucleic acid. Because the virus is so small, it only has just enough room for one type of nucleic acid. And from that one type, it can make the other types of nucleic acid. Now, surrounding the nucleic acid is going to be protein for a virus. The protein, the coat that surrounds the DNA, is called a capsid. The combination of the nucleic acid and the capsid creates a structure called the nucleocapsid. The nucleocapsid is the nucleic acid, be it DNA or RNA, and the capsid protein. The capsid is made up of individual units that are called capsomeres. And the purpose of the capsomeres making the capsid is to protect the nucleic acid, keep it from being destroyed by things like enzymes. So if we zoom in on this image of a virion, we can see that this is a icosahedral virus or a polyhedral virus. And inside of it, we have our nucleic acid. The nucleic acid is only going to be either DNA or RNA. Surrounding it are these little spheres of protein. Each sphere is known as a capsomere. The capsomeres together form a structure called the capsid. The combination of the capsid and the nucleic acid is known as the nucleocapsid. Now, in addition to the nucleocapsid of a virion, there are also going to be these structures called spikes. Spikes are super important because what the spike is, it is a structure that allows the virus to attach to a host. And when it comes to pathogens, the first step of infection is attachment. If you do not have attachment, you do not have infection. So therefore, this is the most important thing when it comes to a virus. This spike attaches to the host, and therefore the spike determines the host range. This is what's happening with the virus COVID-19. The spike has changed, allowing it to spread from, I believe they thought it was bats, to humans, because the spike had mutated to allow it to bind to a receptor that a human has now. So here we see some electron micrographs of naked virions. We'll understand what a naked virion here is in a moment. So what we can clearly see in this first image of mastodinovirus is we can see the icosahedral shape, the polyhedral shape, and you can clearly see in it those capsomeres making up the capsid. What's very interesting about these capsomeres is we don't really understand how they fuse together, how they come together. Some people are furiously working on this, feverishly, not furiously, maybe furiously, but feverishly working on this, so that maybe if we understand how capsomeres self-anneal, well, we can maybe start to make certain types of drugs against viruses that would be almost universal, because all viruses do this. If we look at this transition electron micrograph of the mastodinovirus, now we can clearly see those spikes. It almost looks like, you know, a little stick with a little bit of glue at the end of it, which allows it to stick to its target organism. Now, the virus that we just talked about with its nucleocapsid and spikes is what's known as a naked virus, a naked virion. Now, some viruses are not naked. They're known as enveloped viruses. Enveloped viruses, well, they have an envelope. And what the envelope is, it's plasma membrane taken from the previous host that surrounds the nucleocapsid. Now, not all viruses have an envelope. Only some do. For example, well, the coronavirus has an envelope. The flu virus has an envelope. HIV has an envelope. Herpes has an envelope. Now, what we notice is that on these enveloped viruses, where are the spikes? Here on the naked virus, the spike was directly on the nucleocapsid. But on the envelope virus, the spike has moved from being on the nucleocapsid to being on the surface of the envelope. Why do you think, you can unmute yourself if you want to answer, why do you think it was important for an envelope virus to have its spike on its envelope and not on the nucleocapsid?
Conjugation?
For attachment.
Okay, so for attachment, what do you mean by for attachment?
So it can attach to the host and be able to replicate using the
Okay, so
of the host.
if the spike were to remain on the nucleocapsid of an enveloped virus, would it be able to attach?
No,
Why not?
it doesn't have the spikes.
Well, it has the spike, but it's on the nucleocapsid.
It would be covered by the
covered by
envelope.
the envelope.
Good, Jamie. Exactly.
It would be covered by the envelope.
Excellent. So because the virus now has a envelope, a membrane, the spike must be on the surface of the membrane because it's the surface where the two things are going to interact. The virus interacts with the host at its surface. So here we're looking at an envelope virus influenza. And we can see inside the envelope virus the nucleocapsid. Surrounding that envelope virus is the envelope itself. So here's the nucleocapsid. The envelope is surrounding the perimeter. In between these two things, that's what's known as the matrix. Yes, like the movie Matrix. And on the surface of the envelope, we can see these spikes. And if we zoom in very closely, you can actually see they're very spiky. Almost looks like a brush. Here we see human immunodeficiency virus, or HIV. It too is an envelope virus. We can see its nucleocapsid, the envelope, and the spikes. Now the spikes in this image, they're labeled in green. The receptor that they target is labeled in blue. What do you notice about those green spikes and the blue receptors? Well, if we zoom in here, we can see that the green spikes are attaching to the blue receptors, because that is how a virus attaches to the host. The spike interacts with a receptor. So there are lots of different types of viruses out there. Our job is to organize them so we can understand their evolutionary path, how they're similar to each other, and how they're different from each other. When it comes to the taxonomy of viruses, viral families end in the name Viridae. A viral family is the highest taxonomy of a virus. There are no domains of viruses. There are no kingdoms. There are no phyla of viruses. The highest taxonomical group of a virus is a family. And whenever we have a family name of a virus, it ends in the suffix Viridae. Now, the naming convention of viruses does not follow anything like it would in a living organism. It does not follow Carolus Linnaeus binomial nomenclature. Family of viruses, they follow no consistent pattern. So sometimes the name of the family virus is indicating its appearance, like the Coronaviridae. The Coronaviridae stems from the Latin word corona, which means crown. So the spikes on the coronavirus make it look like it has a crown on. Other names of families of viruses are named after their geographical region, such as Bunaviridae, which comes from the Bunawera in Uganda. We then have the genus. The genus of a virus ends in the suffix virus. And again, there is no naming paradigm. There are no naming rules here. So you can pretty much name it whatever you want. Many times it's again after what the structure looks like, where it's from, or what disease it causes, or what organs it targets. For example, we have this genus, Enterovirus. Enterovirus is a genus of viruses that infect the enteric system. And then we have the species. The species of a virus is often the name of its disease. So for example, poliovirus, which is the name of the species, causes the disease poliomyelitis, or also known as polio. Now, virus species, they do not have to follow that binomial nomenclature. They do not have to be Latinized, and they don't have to follow the genus. In fact, many viral species names are just the common name that we use for them. So let's take a look at two different types of viruses. We have our first type, the herpes viridae, which is the viral family. The genus is herpes virus. The species is human herpes virus, or HHV. Human herpes virus can have different types of strains: HHV1, HHV2, HHV3. These three different strains are named so because they target different areas of the human body, such as the scalp or the skin, such as the genitals or the throat. We then have the other group of viruses, which are the Retroviridae, is a family. Lentivirus is the genus. Again, Viridae is family, virus is genus. And in the case of this species, it's called human immunodeficiency virus, or simply HIV. And again, there are two types of strains here. There are differences in the HIV species. There's HIV-1 and HIV-2. HIV-1 is the predominant HIV found in the world, and it produces the more severe set of disease, while HIV-2 is less dominant and produces a less severe disease. When we often talk about viruses, we often refer to them informally. We don't use that binomial nomenclature. We don't really follow the same rules when it comes to living cells because they're not alive. So we can group viruses together based on things like sharing routes of infection. These can be completely unrelated viruses, but they're just named so because they do something similar, such as the enteric viruses. The enteric viruses, there's a bunch of viruses that fall under the enteric viruses, not all closely related. They're not all part of the same family, not all part of the same genus. And the way that we group them together is that they use the same route of infection, oral fecal route. We then have respiratory viruses. They use the respiratory routes. We can group them together because they originate from animals. When a virus originates from animals and infects a human, we call that a zoonotic virus because it causes a zoonosis. A zoonosis is when an infection comes from an animal and infects a human. And this always equals pandemic or epidemic. That's why it's bird flu, swine flu, the bat coronavirus. Those things always lead to devastating diseases. Another way we can informally group together viruses are by what is spreading them, such as arboviruses. Arboviruses, or arthropod-borne viruses, they are spread by arthropods, such as mosquitoes, ticks, and they can cause diseases like yellow fever, dengue fever, West Nile encephalitis, La Crosse encephalitis. These are not related viruses. It's just that we tend to group them together because they share something similar, how they are being transmitted. So here we just have a list of viral groups: enteric viruses, respiratory viruses, sexually transmitted viruses, zoonotic viruses, the routes in which they're spread, and the types of viruses that there are, like enterovirus, norovirus, rotavirus. These are not, they're not related to each other. But the reason we're grouping them together is because they follow the same mechanism of transmission. All right. I think this is a good place to stop. What I'm going to do before I stop the lecture is I'm going to open it up to questions. So if anybody has a question, you can unmute your microphone. Please make sure it's quiet in the background, or type a question into the chat window and we'll answer it for you.
Hi. Can we go back to the viruses that have the envelope and the ones that don't have the envelope?
Sure. Oops. All right, so what do you want to know here?
I guess, how is it different when it has the envelope versus when it doesn't? Is the envelope like a layer of protection for the nucleocapsid, or?
So the envelope is an artifact of how the virus gets out of the host cell. In fact, an envelope is not a layer of protection. Envelope viruses are actually more sensitive to chemicals like soap than naked viruses.
Okay.
So it actually has to deal with how the virus escapes the host. When naked viruses escape, they typically escape by blowing up the host. So the host cell lyses.
Oh, okay.
That's not good for the virus because now it no longer has a host to make more copies of itself. But an envelope virus, it buds off the host cell. So the host cell actually stays alive, so it can continually produce viral particles.
Oh, and when you say it stays alive, like that's when like something like HIV or something similar to that, it's kind of invading it, and that newly infected cell is reproducing itself rather than having to. It does it do it some? Does it like would it work in a different way if it didn't have the envelope?
Well, it would probably lyse the infected cell.
Okay.
So if you lyse the infected cell, that means that that viral factory that's producing viruses is no longer able to produce viruses.
Got it. Okay.
But if the host cell stays intact, well, it can continually produce viruses as long as that host cell stays alive.
Thank you.
I actually asked a question on the chat about that. So it means like the naked virus is more harmful than the envelope virus, right?
Now, when you say harmful, you're implying the severity of the disease.
Yes.
The virus's composition does not affect the severity of the disease. It's the type of virus and the host and what organs are targeted that affect the severity of the disease. So what you're really trying to say here is, are envelope viruses more stronger than non-envelope viruses or naked viruses? Meaning, are they more resilient? No. Naked viruses are stronger in that they can more likely survive disinfection, drying out, dehydration, than an envelope virus, because the lipids are very sensitive. And as soon as you destroy those lipids, what goes away with the lipid membrane? Those spikes.
Mhm.
If you get rid of the envelope in an envelope virus, the spikes go away as well. And what were the spikes for? The spikes were for targeting the host cell. So now this basically envelope-less envelope virus can no longer target the host cell. So naked viruses are more resilient than envelope viruses.
That makes sense to me, but so like in the cases of herpes and HIV, like it has to do with other reasons why we don't really have a cure for it, not so much it being an enveloped virus.
So the reason we don't have a cure for things like HIV and herpes is because HIV and herpes are retroviruses. And what that means is that they take their RNA, which is their genome is based in RNA, and they convert it into DNA. And when they convert it back into DNA, they incorporate that DNA into your host cells. So literally the virus becomes a part of your chromosome. So therefore, the only way to actually get rid of it is to cut it out of your chromosome. And we're only just approaching the medical technology to do that with things like CRISPRs. Otherwise, it's impossible.
Okay, thank you.
Mm-hmm.
I have a question. If the virus itself lyses inside the envelope, does it still act the same?
So the virus doesn't lyse. It's the cell that lyses, because lysis means to break open a cell. The virus doesn't have a cell, so it can't lyse. So I'm not 100% sure what you mean.
Okay.
Can you just rephrase it in some? You're saying here, if the virus lyses in the envelope, you mean if the envelope breaks?
No, I was referring to the nucleus capsid. Can that lyse itself or no?
No. What could happen is it can degrade.
Okay.
If the nucleocapsid is defective, then it won't act the same. It'll still grab a hold of the target cell because the spikes are still available, but it itself probably won't be able to replicate. So it's essentially a dead virus.
Okay.
All right. So I'm going to end the lecture here. I do really appreciate all of you working with me during this very difficult time. Usually people have an entire semester to prepare for online instruction. We're doing it in a matter of days. So I do understand that there may be issues that come along. We did have some issues today during the exam, but I was able to fix those. I'm sorry, Fatima, you said pillis. What were you saying there? Or was that from a while ago?
That was from a while ago. I was asking about the attachment of, I don't even remember.
That's fine. That's fine.
Is a spike kind of like a pillis?
Right.
Kind of, yeah. It's kind of like a pillis or a fimbriae, where it attaches to a specific place. Yeah. So you really got to think. What we really have to start thinking about a cell is not like, it's not like a body. Cells are like machines, and they're looking for specific things that they attach to by shape. So just like the fimbriae of Neisseria gonorrhea looks for things like mucous membranes, the enteroviruses look also for mucous membranes because they have a very specific receptor that they attach to.
I remember now. I was referring to the picture with the virus that had the green surrounding it and the cell having blue. And I was asking, is that like a pilus of a sort?
No, those are the spikes.
Okay.
All right, so if we don't have any other questions, I'll end it here. Let me end the recording. We are going to cover prions later, yes.
Professor, are you going to post a review for the lab exam?
I will, yes, I'll do that. Please make sure to look at your quizzes. I'll post the quizzes that you have taken, but I haven't given back yet. I'll post those onto Blackboard by sometime early this week. Okay.
Also, will you post the audio for this lecture?
Yes, I will be sending you the link.
Okay, thanks.
You're welcome.